Hello everyone. In this video, we will learn about the staining processes, the major staining process which is the Gram staining process. We will discuss that in detail. Also prior to it, we will learn that what are the staining dyes and its type and how they react with the biological components to give a particular stain. So let's begin. So what is staining? Staining is a biochemical technique of coloring the specimen. Of course, when something is colored, it is easily differentiable from the others and it is easy to view them in microscopes as well. So different types of dyes are used for staining. These are organic compounds which contains two functional chemical groups. One group gives dye its characteristic color. The other is the ionizable chemical structure which helps to solubilize the dye and facilitate its binding to different cellular structures. So there can be various types of dye. The two major are the cationic dye. These are the basic dyes that react to the groups that have negative charges on them. For example, the crystal violet, methylene blue, safranin, malachite green. The other is the anionic dyes. These are the acidic dyes that react to the group that have positive charges on them. Examples include eosin and fuchsin. So why do we need to stain the cells? To differentiate them from their surroundings. This is the simple staining process. To differentiate a particular cell type from another cell type, the differential staining process, which includes the Gram staining and acid-fast staining. To identify some specific cellular structures like the flagella or the endospores, these are the specialized staining processes. So the differential staining process, the Gram staining, that actually differentiates the Gram positive and negative bacteria, we will learn about it in a bit detail. So what is Gram staining? It is basically a staining procedure and which is used to differentiate the Gram positive and Gram negative bacteria. So before we move ahead, we should first know some basic thing about the Gram positive and negative bacteria, which is basically about their cell wall and its composition and its abundance and structure as well. So here we can see that the cell wall of Gram positive, this brown structure, it is made up of peptidoglycan, which is also present in the Gram negative. But when one can easily identify that the Gram positive cell wall is much more thicker than the Gram negative one. Also, the Gram positive cell wall, the peptidoglycan, is the outermost layer, whereas in Gram negative, above this peptidoglycan layer, there are other outer membrane structures. These are the lipopolysaccharide and protein composed of structures. So basically, the dyes have to react with this cell wall. They have to penetrate into the cell wall so as to show the colors. But in Gram negative bacteria, this cell wall is not readily available. The dye has to cross this outer membrane. Also, the cell wall which is available is very thin, so the dye is not able to readily react with it. Whereas in Gram positive bacteria, one can see that it is easily available. The cell wall is easily available. It is abundant. So most of the dye can penetrate into it and can actually show its colors. So basically in Gram staining, we actually make our dye react with the cell wall and make the cell wall colored. And this cell wall, when it gets colored, it's actually easy to be viewed. So in Gram staining, the Gram positive bacteria basically are actually colored and they are differentiated from the Gram negative bacteria in a way. So something in detail here. The Gram staining is a differential staining method that differentiates the bacteria based upon the physical properties of their cell walls. The technique is named after its inventor, the Danish scientist Hans Christian Gram, who developed this technique in 1884. The technique has 4 basic steps. The one is crystal violet staining, the iodine staining, the alcohol acetone decolorization, and the safranin counterstaining. So one can remember this word CIAS, CIAS, to easily remember the step. C for crystal violet, I for iodine, A for alcohol or acetone, and S for safranin. The first is the crystal violet primary staining. The bacteria is first stained with the basic dye crystal violet, so it is a basic dye. It is a primary stain and imparts purple color to all the cells. The next is the iodine treating with it. Then the cells are treated with Gram's iodine solution. This allows the stain to be retained better by forming an insoluble crystal violet iodine complex. So this complex is insoluble. Iodine is used as a mordant. What is a mordant? It actually helps in increasing affinity of a particular stain to a biological specimen. Here in this case, the crystal violet. So iodine is a mordant in this case. So you can see that when you apply the crystal violet, all the cells in the culture or the specimen, it gets its purple color. And to increase the strength of this purple color, we again add the iodine so that this binding is much more stronger. So iodine is a mordant here. Okay. A for alcohol acetone decolorization. Gram's decolorizer is a mixture of ethyl alcohol and acetone. It is then added upon the specimen. This removes the stain from the specimen. So this is done to remove the stain. This is the differential step. So Gram staining is a differential staining procedure, and the differential step in this Gram staining procedure is the differential step of alcohol acetone decolorization. As after this step, some bacterial cells are purple in color and some are colorless because they lose the stain. And which cell lose the stain? In which the binding is not very proper, the Gram negative ones. So the bacteria that retain their purple color are classified as Gram positive bacteria, and the other ones are the Gram negative bacteria. But then we are not able to see these Gram negative bacteria via microscope. So again, we need to counter stain it with some other color so that we can easily differentiate between the Gram positive and Gram negative ones. So we already know that the Gram positives one are purple in color. So let's color the Gram negative ones also. So then what we do is S for safranin counter staining. We apply this safranin to the culture or the specimen, and the Gram negative bacteria which were invisible previously are now easy to see. This basically does not affect any Gram positive bacteria which were already stained, because there is a very strong bond, insoluble complex has been formed already in their cell wall, the crystal violet iodine complex, and the safranin doesn't affect the Gram positive bacteria. So then the alcohol counter staining can be seen here, and then the safranin can also be added to view the Gram negatives. So here we can see this is a slide showing the Gram negative and the Gram positive bacteria. One can remember the simple thing like P for positive and P for purple. So Gram positives stains actually stain purple in the Gram staining. So this is a trick to remember: P for positive, P for purple. And here are some examples of the Gram positive and negative bacteria. So the major example includes for positive bacteria Staphylococcus aureus, then the Bacillus, then Streptococcus pneumoniae. Gram negative bacteria, the major example Vibrio cholerae, the most used one this Escherichia coli, okay, and many more. So one can remember this, and I hope you liked the video. Don't forget to like, share and subscribe to the channel. Stay tuned for more such updates. Thanks for watching.